Let's get this party going.
Jump in the Cadillac, girl, let's put some miles on it. Anything you want.
So the topic of today is going to be neurotransmitters, and the specific one I'm going to talk about is acetylcholine. To start off, it's probably helpful to describe what a neurotransmitter even is. To put it simply, a neurotransmitter is a messenger that sends a signal from one neuron to another. It can also be described in terms of synapses. The synapsis refers to the area between the presynaptic neuron, which refers to the neuron where the neurotransmitter is actually coming from, and then the postsynaptic neuron, the neuron receiving the signal. So the neurotransmitter really functions like its name. It transmits a signal and allows communication between all the neurons in the body. So let's begin with acetylcholine. The main hub for acetylcholine is in the brain and the spinal cord, which we can refer to as the CNS or the central nervous system, and some tissues in the peripheral nervous system or the PNS, but this is not like its main area. That should be very importantly noted. There are, obviously it's there in some tissues, but it's not the main place. There are two main neurotransmitters that occupy the PNS, and that is epinephrine, or epi for short, and I can't say it, no epinephrine, no, any is the other one. Anyway, so I'll talk about those two probably in a later video. Acetylcholine's functions are mainly associated with memory and functions in the skeletal muscle, the cardiac muscle, and then some glands. So let's talk about how it's synthesized. If you take the word apart, you can see that the second half is choline, right? So this is an indicator of what acetylcholine is made of, choline. Choline is a nutrient that we get from foods we eat, like vegetables, fruits, and dairy. You don't really ever have a deficiency in choline, like it's not really common. So how acetylcholine made begins with choline being absorbed into the axon terminal of the absorbed into the axon terminal of the neuron by the enzyme choline acetyltransferase, okay? Which essentially sits on the neuron and just is ready to take in choline at any moment. Inside the terminal, there's another enzyme called acetyl-CoA, which alongside with choline, voila, makes acetylcholine. So now what? Well, through a process called exocytosis, acetylcholine is released into the synaptic cleft, which is just a fancy word for like the area between the two neurons. So now it's in a place where it's ready to bind to the receptors of the postsynaptic neuron and cause a response. Sorry, I'm literally like looking at my notes and then doing this at the same time. So it's like, there are actually two different receptors acetylcholine can bind to, and those are the nicotinic receptor and the muscarinic receptor. So first we're going to talk about the nicotinic receptor. When acetylcholine binds to nicotinic receptors, this causes an ionotropic response, aka ion channels are opened, mainly positively charged ions like sodium. This can be thought of as like an excitatory response, and here's why. Because these ions are positive, they make it easier for the postsynaptic membrane to reach threshold, which is -55 or -50 depending on what you're reading online. -55 millivolts, and thus action potential. I'm not going to explain threshold and action potential because if you're watching this video at this point, you should know what those things are. Anyway, so this rush in positive ions is exactly the characteristic that describes EPSP, or excitatory potential. Nicotinic receptors' ability to have direct contact with these ion channels leads us to the conclusion that they are inherently faster, or faster than muscarinic receptors at least, which let's talk about those now. Muscarinic are the slower receptors and are also called metabotropic. So what does this mean? This means it has a second messenger, or in a way a second receptor kind of, within the postsynaptic membrane that activates a series of events that have to be considered after G proteins are activated, which G proteins are associated with muscarinic receptors. So the important thing really to take here, I know it can be kind of confusing, but the important thing is that unlike ionotropic receptors, muscarinic receptors are not directly linked to ion channels, making them slower by default. On top of this, when G proteins are activated, there is variation of whether an excitatory or inhibitory response may come about, which could either drive the neuron away or closer to threshold. Another difference between the two receptors is that nicotinic receptors cause short-lived responses that are short-lived and immediate, while muscarinic receptors are longer and usually more widespread. So that was annoyingly long. So what happens after the neurotransmitter is done doing its deed on the receptor? Well, this is actually where acetylcholine stands out as a neurotransmitter, as it is the only neurotransmitter that functions this way. Acetylcholine is actually broken down in the cleft by an enzyme called acetylcholine esterase in two parts, acetate and choline. So acetate diffuses away, which I'm not really sure where it goes, so don't ask because I don't look it up, I don't know. And choline then repeats the process of re-entering the presynaptic neuron, although I believe it's usually in a smaller amount because it's been broken down, but I mean it doesn't really matter because we get choline anyways from like when we eat, so it's whatever. So yeah, that is how acetylcholine works. I would recommend taking a look at drugs like benztropine, which is an anticholinergic, and donepezil / I can't say that one, rivastigmine, which those two are acetylcholinesterase inhibitors, and try to explain how they function based on these pharmacological categories they're put in, because based on the info that I just showed you, you should be able to figure out how these drugs function in the neuron cell, considering they're an anticholinergic or acetylcholinesterase inhibitor. So yeah, I can't believe that took me 10 minutes. Thanks for watching.